why do we age in the first place, right? So we have the Greenland, we have the, bowhead whale lives to 200 years old. We live, the Greenland shark lives to 500 years old, has pups at 200 years old, and we make it to 100 on a routinely regular basis. But why do we age, and why were there no factors selecting for us to live longer than that?
Yeah. Well, the first answer to the second question is there was no need. And evolution doesn't build things that there's no need for. And on the savannah, we were likely to die from war or infection or starvation within 50 years. So living to 100 was really not helpful. The reason why we age is debated among academics, but what I strongly believe, based on the evidence that my lab's been garnering, 25 years now, is that it's not just wear and tear. We're not just breaking down. There aren't just things going wrong, but it's all about information and entropy.
Yeah.
And that what keeps us young is that we have great information. We've got our pristine genome and our pristine epigenome, which are the structures that read the genome. And those two elements of information, they get lost over time due to noise. And we've identified some of the causes of that noise. DNA breaks of chromosomes is part of that noise. Stress on a cell. If you crush a nerve cell, it will accelerate its aging, we showed in a 2020 Nature paper. And so I think of the body not like a lump of meat now, I think of it like a computer, and the software gets corrupted over time. And then the great news is we found that there's a backup copy of that software that can be reset.
Amazing. You've not done any podcasts in the last few years, so I'm super happy to have you here. So I really want to go into what you've learned. So the information theory of aging, you published that as part of your book Lifespan. Time did an amazing piece on you and the information theory of aging. So I was so happy that Mark Benioff, who owns Time, did that. I was like, yes, it's good to get the exposure for this. So let's take it a step further in explaining what that information of what that aging theory means. And have you had any new proof points supporting that in the lab?
Yeah, we've come a long way since 2020.
So just to settle things, you get 3.2 billion letters from your mom, 3.2 billion letters from your dad. That's your genome. It doesn't change over the course of your life. So as I like to ask people, like, why do you look different at 20 and 100? Why don't you look the same? And that genome codes for, what's the current estimate? 22,000 genes?
Yeah.
Thereabouts. So epigenetics is effectively which genes are on and which genes are off, right? As you age, if I just have this right, as you're aging, the wrong genes turn on and the wrong genes might turn off, and that's causing some of this aging effect.
I can't add a lot to that. You summed up the theory well. And I did put it in Lifespan, the book. So if anyone hasn't read Lifespan, it's all in there. And book 2, hopefully next year, will come out. But it was a risk putting it in that theory and all of our results into the book because we hadn't published it yet.
Yeah.
But since then, since 2020, when the book came out, we have published a couple of big papers, one in Nature and one in Cell. And it's really strong evidence now that the theory
is real.
Stands up. What have we learned since? Well, we're trying to figure out how is it possible to reset a cell and get back the original structures of the epigenome. And if we were to dive into the cell, okay, let's say we're going to pierce the membrane, go inside a cell, then you swim down and then you see the nucleus. There's another membrane. There are little pores in the membrane. We can swim through those. Now we can see the chromosomes. And that's really the epigenome. And if we unwrap those chromosomes, what we would see is the 6 feet of DNA that's in the cell, of every cell. But the DNA is wrapped up in proteins. And that wrapping is important for 2 reasons. One is that's how you fit 6 feet into a microscopic cell, but also whether it's wrapped tightly or not determines whether a gene is on or off. And if it's tightly bundled, we call that silent chromatin or heterochromatin. And a lot of the genes you want to be switched off because you don't want a liver-specific gene on in the brain and vice versa. So there are these bundles of DNA and loops of DNA.
And when it's bundled tightly, it can't be read. It can't be transcribed.
Because it's hidden. Exactly.
Yes.
It's tightly bundled. And actually, one of the big clues that this was relevant to aging was when I was in the lab of Lenny Guarenti as a 25-year-old postdoc. And as a team, we uncovered that there were these silent information regulator genes called the sirtuins, and their job is to bundle up DNA.
Mm-hmm.
And at the time, we're thinking, what the heck has bundling DNA got to do with aging? We were expecting to see antioxidant genes and telomere extending genes, but we found these silent information regulators. And that's why information has been key to my research ever since. But getting back to what happens during aging is that these bundles and these loops, they change over time, in part because the cell loses the ability to bundle them and you get these loops open up. But also because the epigenome is changing all the time. Our genome is stable. We don't change our genome much. And in fact, mutations are not as abundant as we thought. And you can actually, I could take one of your cells and clone you. And even though your cells are old, we can start new life with those cells.
I love that, right? And so I know one set of experiments were done actually took cells from 23 sequential mice clones that showed, you could take a cell, clone the mouse, grow it out, take a cell, clone the mouse, grow it out. And I think they got bored and stopped at the end
and
of
they're still healthy, right? These mice aren't getting old.
Yeah.
But if the old theory is correct, that mutations cause aging, then you just end up getting older and older babies.
Right.
That doesn't happen.
Yeah.
So therefore, I'm very convinced so far that the epigenome is the issue, because going through sperm and an egg and fertilization resets the epigenome. So you can start life again. Mm-hmm. But the good news is we've figured out a way to safely reset that without having to clone yourself. Mm-hmm. So think of it like that, as resetting the system, like going through conception without having to be reborn.
Amazing.